tissue systems. Then tissue systems, three types. One is dermal tissues, second one ground tissues, and third one vascular tissues. Three types. So first one, dermal tissues. Different theories. According to histogen theory, dermal tissues develop from dermatogen. According to 𝗂𝗽𝗃 theory, dermal tissues develop from tunica. And even meristem, protoderm, it gives epidermis. So different types of meristems, they gives dermal tissues. Then dermal tissues includes epidermis, then unicellular or a multicellular outgrowths. It includes hairs, trichomes, and even stomata. These are dermal tissues. So first one, epidermis. Epidermis, common structure. Epidermis is commonly unilayered. It is unilayered, cells closely arranged without any intercellular spaces. Then cells rectangular or barrel shaped, and the cells are living cells. They contains protoplast and vacuoles. Generally, this epidermis contains leucoplast, colorless, but particularly in hydrophytes and 𝗖ophytes, they are shade-loving plants. In this, that epidermis contains chloroplast. Epidermis contains chloroplast. Now you can take root epidermis, stem epidermis, and leaf epidermis. If you take root epidermis, in root epidermis, stomata and a cuticle both are absent. Then root epidermis generally unilayered, but exceptional. In epiphytes, rightly, root epidermis is multilayered. Epiphytes, root epidermis is multilayered, dead and hygroscopic, called velamin tissue. Then this root epidermis contains two types of cells, smaller and larger cells. The smaller cells, in these smaller cells, they develop unicellular outgrowths, rightly These unicellular outgrowths are called root hairs. They are extensions of epidermis. And cells developing root hairs are called trichoblast. Called a trichoblast. Because of root hairs, root epidermis is called epiblemma or rhizodermis or piliferous layer. This is about root epidermis. And root hairs, they absorb capillary water by a process called osmosis. Then velamen tissue, it is multiple root epidermis. It absorbs water by imbibition. The next one, stem epidermis. Stem epidermis generally unilayered. Then cells rectangular or barrel shaped, contains vacuolated protoplast. Then stem epidermis, it contains stomata and also cuticle. Then generally stem epidermis contains multicellular outgrowths. These multicellular outgrowths are called trichomes. This is root epidermis, then stem epidermis. Next one, leaf epidermis. Only leaf epidermis present on both sides of leaf. Upper side called upper epidermis or adaxial. Lower side called lower epidermis or abaxial. Then same, both epidermis, living cells, barrel shaped, contains vacuolated protoplast. Generally, leaf epidermis is also unilayered, but in some plants, this leaf epidermis is a multi-layered, multiple leaf epidermis present in ficus, peperomia, and even nerium. Multiple epidermis. In some plants, this leaf epidermis contains silica crystals. These silica crystals present in grasses. That rough texture of grasses due to these silica crystals. In some plants, this leaf epidermis contains myrosin enzyme. Example, brassicaceae. In some plants, this leaf epidermis contains calcium carbonate crystals called cystoliths, and a cell containing cystolith is called lithocyst. Example, ficus, the family Moraceae. Then in grasses, upper epidermis contains hygroscopic cells. These hygroscopic cells are called bulliform or motor cells. They are flabelliform, means a fan shaped. Turgor changes in bulliform cells leads to rolling and unrolling of leaf lamina. So this is about epidermis, separately root epidermis, stem epidermis, and leaf epidermis. Next one, stomata. So first one, structure of stomata. The stomata contains Turgor operated valve. This Turgor operated valve is called stoma. The stoma is surrounded by green epidermal cells. These green epidermal cells are called guard cells. These guard cells in dicots, reniform or a kidney shaped or a bean shaped. In grasses means monocots. Monocots guard cells are dumbbell shaped. Then these guard cells are surrounded by non-green epidermal cells. These non-green epidermal cells are called subsidiary cells or accessory cells. Stoma, guard cells, and subsidiary cells together called stomatal apparatus. Then walls of guard cells shows uneven thickening. Inner walls of guard cells surrounding stoma are thick walled. Outer walls of guard cells towards subsidiary are thin walled. Rightly, function of guard cells. They are responsible for opening and closing of stomata. It is structure of stomata. Then types of stomata based on distribution. First one, apple and mulberry type. Apple and mulberry type means stomata confined to only lower epidermis. If stomata confined to only lower epidermis, called apple type. Example, apple and mulberry. Second one, potato type. In potato type, stomata unequally distributed, but more stomata confined to lower epidermis, called potato type. Example, same potato. Next one, oat type. Oat type, stomata equally distributed. Stomata equally distributed, called oat type. Rightly, it is described as amphistomata type. Amphistomata. The next one, water lily type. Water lily type, in which stomata present only on upper epidermis, called water lily type. Next one, Potamogeton type. In this type, stomata absent. If it is present, it is vestigial or non-functional. This non-functional stomata present in submerged hydrophytes. Example, Potamogeton, Vallisneria, even Hydrilla. These are types of stomata based on distribution. Water lily, common in hydrophytes with the floating leaves. Water lily means nymphea. Even Neelambo. Neelambo is lotus. And Victoria. Importance of Victoria, largest leaf. Common name, Amazon lily. These are based on distribution. Stomata includes subsidiary cells and guard cells. If subsidiary cells and guard cells, if both are developed from same other cell, called synditochelic. If both are developed from different cells, rightly called haplochelic. and common information. If stomata present on only upper epidermis, it is called epistomata. If stomata confined to only lower epidermis, called epistomatas. If stomata confined to lower epidermis, it is called hypostomata. Then generally in xerophytes, stomata confined to lower epidermis called hypostomata, and this stomata is deeply seated, that is called sunken type. Next one, epidermal outgrowths. Epidermal outgrowths, two types: epidermal hairs and emergences. Epidermal hairs developed only from epidermis, called epidermal hairs. These are two types: hairs are trichomes and scales. Generally, these hairs are trichomes. They are unicellular, commonly unicellular or multicellular, uni or multicellular. If they are multicellular, they are uniseriate. Uniseriate means one row of cells, or they are multiseriated. Multiseriated means many row of cells, multiseriate. So hairs are trichomes: unicellular, multicellular, uniseriate, multiseriate, unbranched or branched, non-secretory or secretory. If they are secretory, called glands. Best example for glands: stinging hairs in urtica, which secrete poisonous substance. And in some plants, these multicellular outgrowths are tree-like. If tree-like, called dendroid. Example, Withania. In some plants, these are star-shaped. If star-shaped, called stellate. Stellate generally common in Solanaceae and Asteraceae. These are hairs are trichomes. Then second one, scales. Scales means actually they are multicellular. Scales are multicellular, then with the stalk and attached to epidermis, called scales. So these are epidermal hairs. They develop only from epidermis. Then next one, emergences. Emergences means they develop from epidermis and its underlying tissue, called emergences. Emergences Example, prickles in a rose. Ground tissues means that the tissues present in between epidermis and a stele called ground tissue, and some tissues also present within stele. That is why ground tissues are divided into two types. One is extrastellar ground tissue. Second one, intrastellar ground tissue. Extrastellar ground tissue, it includes ectodermis, hypodermis, general cortex, and endodermis. These are extrastellar ground tissues. So, first one, ectodermis. Ectodermis is character of roots. Ectodermis present in roots. Actually, in roots below the epidermis, parenchyma cells present, but gradually, due to deposition of suberin, they become thick-walled and forms ectodermis. Then, function of ectodermis. Ectodermis is suberized, so it prevents that absorbed water to exit outside the root. Not throughout the part ectodermis present. Only in matured region ectodermis present. In root hair region, ectodermis is absent. So it is character of roots. The next one, hypodermis. Hypodermis, character of stems. In stems, hypodermis is present. That too, hypodermis present in dicot stem and also monocot stem. In dicot stem, hypodermis is collenchymatous, made up of collenchyma. This collenchymatous hypodermis forms continuous ring or homogeneous in sunflower. And this collenchymatous hypodermis occurs as patches in cucurbita. So, collenchyma means living mechanical tissue because it provides mechanical support and it is living tissue. So, young stem, in young stem, this collenchyma contains chloroplast, so help in assimilation. In monocot stem, this hypodermis is made up of sclerenchyma. It provides only mechanical support. Only mechanical support. Then, next one, general cortex. This general cortex present in both stems and roots. Present in stems and roots. First in stems, this general cortex means it is present from general cortex, present from or in between hypodermis and endodermis. Hypodermis and endodermis. So, in between that layer or tissue present in between hypodermis and endodermis is called general cortex. This general cortex predominantly parenchymatous. So, parenchymatous means cell spherical or oval with large intercellular spaces. Outer cortical cells contains chloroplast, so help in assimilation, and inner cortical cells contains leucoplast, so help in storage. And this general cortex present only in dicot stem, absent in monocot stem. Now, general cortex in roots. In roots, general cortex present in between exodermis and endodermis. So, portion in roots, portion in between exodermis and endodermis is called general cortex. This general cortex, no doubt, parenchymatous, so cells oval or spherical, then with a large intercellular spaces. In roots, general cortex perform two functions. One is this, it help in lateral conduction, that is mostly apoplastic pathway, and along with conduction in roots, this general cortex contains leucoplast, so it help in storage. So both roots contains general cortex. In both roots, they help in lateral conduction and also that storage. But in stems, general cortex present only in dicot stem and absent in monocot stem. The next one, endodermis. This endodermis present in stems and roots. And point, innermost layer of cortex. Innermost layer of cortex is called endodermis. Generally, in stems, endodermis is not well developed. That too, endodermis present only in dicot stem and absent in monocot stem. In dicot stem, this endodermis stores starch grains. So, in dicot stem, that endodermis is called starch sheath. In monocot stem, it is absent. Then roots. In roots, endodermis is well developed. Some of the endodermal cells, generally endodermal cells means unilayered, made up of barrel-shaped cells and cells closely arranged. So intercellular spaces absent. Particularly in roots, some of the endodermal cells, particularly in roots, some of the endodermal cells shows tangential, these are tangential, and also radial thickenings. These thickenings are due to lignin and suberin. Such ligno-suberin thickenings are called Casparian thickenings because first reported by Caspary. So because of Casparian thickenings, this endodermis forms barrier between cortex and stele. It means this Casparian thickening is impermeable to water. That is why some of the endodermal cells, some of the endodermal cells exactly opposite to protoxylem are thin-walled. These thin-walled cells are called passage cells or called transfusion cells. Passage or transfusion cells. They permit water. So, these are extrastellar ground tissues, exodermis in roots, hypodermis in stems, general cortex, then endodermis, present in both stems and roots. Then, intrastellar ground tissues. Intrastellar ground tissues means within stele. It includes pericycle, medulla, medullary rays, and conjunctive tissue. Pericycle, medulla, medullar rays, and conjunctive tissue. So, first one, pericycle. Pericycle means outermost layer of stele. Outermost layer of stele is called pericycle. First, pericycle in stems. Below the endodermis, there is pericycle. Generally in stems, pericycle is multi-layered. In stems, pericycle is multi-layered and sclerenchymatous. It is multi-layered, totally sclerenchymatous and homogeneous in cucurbita. In sunflower, this multilayered sclerenchyma is formed by both sclerenchyma and parenchyma, and generally this pericycle, it forms patches over phloem. Rightly, it is called bundle cap. So, in stems, pericycle, multilayered and sclerenchymatous. It is totally sclerenchymatous and homogeneous in cucurbita. In sunflower, multilayered, formed by both sclerenchyma and parenchyma, but it is discontinuous. It occurs as patches over that phloem, so rightly called bundle cap. Then pericycle absent in monocot stem. So, in monocot stem, general cortex, endodermis, pericycle are absent. Now in roots, generally pericycle is unilayered. Some plants in roots pericycle is multilayered. But in roots generally pericycle is unilayered. Pericycle is unilayered. It is parenchymatous. Parenchymatous in both roots, and in both roots this pericycle produces lateral roots. So that is why lateral roots are called endogenous. Lateral roots are endogenous. Reason, they develop from pericycle. Then, now point, this pericycle is throughout life parenchymatous in dicot root. In dicot root it performs two functions. One is it produces lateral roots. Then it undergoes dedifferentiation and it produces cambium in dicot root. In dicot root, more 𝗂𝗃 and less vascular cambium formed from pericycle by dedifferentiation. Monocot root, it produces lateral roots. Young monocot root, pericycle is parenchymatous, but in old monocot root, this pericycle becomes sclerenchymatous. Sclerenchymatous means dead tissue. So no dedifferentiation. So cambium absent in monocot roots. So no secondary growth in monocot roots because no cambium. Only in dicot root that secondary growth takes place because cambium produced from pericycle. This is about pericycle. The next one, medulla. Medulla means generally medullary cells parenchymatous. Generally medullary cells parenchymatous. In dicot root, medulla is either reduced or medulla is absent because during xylem differentiation, these metaxylem vessels reaches to center. So medulla is reduced in a dicot root. But in monocot root, medulla is enlarged because metaxylem vessels, they present at periphery. So monocot root, medulla enlarged. Dicot root, medulla is reduced. So it is monocot root, medulla enlarged. Then in monocot root, young monocot root, medulla is parenchymatous. But old monocot root, this medulla contains or it is sclerenchymatous, provide mechanical support. These are in roots. Then dicot stem. In dicot stem, medulla is enlarged because in dicot stem, vascular bundles arranged in a ring. So in dicot stem, that medulla enlarged. Monocot stem, medulla is absent because in monocot stem, vascular bundles scattered, so absent. Then next one, medullary rays. These are vascular bundles Medullary rays means they present only in dicot stem. In dicot stem, some of the medullary cells extend in between vascular bundles, so called medullary rays. These medullary rays present in dicots and also gymnosperms. Particularly in dicot stem, medullary rays help in radial conduction, means lateral conduction. And at the time of secondary growth, these medullary rays produce interfascicular cambium by dedifferentiation. By what? Dedifferentiation. So, dicot stem contains medullary rays. Monocot stem, medullary rays absent. Among roots, in dicot root, medulla is totally reduced. Then monocot root, medulla is enlarged. No confusion. It means if question reduce medulla, dicot root, monocot stem. Enlarged medulla, dicot stem and monocot root. Only in dicot stem, medullary rays present. Dicot stem and gymnosperms. Particularly in dicots, they produce interfascicular cambium by dedifferentiation. Instead of medullary rays, in roots, conjunctive tissue is present. Conjunctive tissue means parenchyma. Medullary rays also parenchymatous, but present in stem and in roots, these parenchymatous cells which connects adjacent vascular bundles. This is rightly called conjunctive tissue. Conjunctive tissue present in both roots, but in dicot root, conjunctive tissue by dedifferentiation produces cambium. It means in dicot root, cambium is produced from both pericycle and conjunctive tissue. In monocot root, pericycle is sclerenchymatous. Even matured root, pericycle is sclerenchymatous. Even conjunctive tissue is sclerenchymatous. They provide only mechanical support. Your point is sclerenchymatous parts in monocot root, medulla, conjunctive tissue and even pericycle. These are extrastellar and intrastellar ground tissues. The next one, ground tissue in leaf. The point is, ground tissue of leaf is called mesophyll. So, ground tissue of leaf is called mesophyll. Mesophyll is predominantly parenchymatous. Mesophyll is parenchymatous. Then cells generally isodiametric or elongated, columnar, contains chloroplast. So, main function of mesophyll, it is photosynthetic. But in dicot leaf, mesophyll is differentiated. Mesophyll is differentiated into, one is palisade parenchyma. This palisade parenchyma present towards adaxial or upper side. Here cells columnar, closely arranged and without any intercellular spaces. And this palisade contains chloroplast but present at periphery and perform photosynthesis. And second one, sponge parenchyma. The sponge parenchyma it is towards abaxial side. Phallic sponge towards abaxial side. Sponge cells loosely arranged and large intercellular spaces present. They also contains chloroplast, so help in photosynthesis. But photosynthesis is not main function. Storing air. Besides storing air, they perform photosynthesis. So in dicot leaf, mesophyll is differentiated into palisade and sponge parenchyma. In grass leaf, mesophyll is undifferentiated. Entire mesophyll is made up of only one type of cells, that cells columnar or cylindrical, and they contains chloroplast. Perform photosynthesis. So differentiated mesophyll present in dicot leaf. Undifferentiated mesophyll present in monocot leaf. Now, third type is vascular tissues. Third type is vascular tissues. Vascular tissues means they include xylem and phloem. Vascular tissues include xylem and phloem. They occur in the form of bundles. So, rightly called vascular bundles. Here types of vascular bundles: radial, then collateral, then bicollateral and concentric. Four types of vascular bundles. So first one, radial vascular bundles. Radial vascular bundles, characteristic feature of roots. Only in roots, vascular bundles are called radial. Radial means xylem and phloem separate. So xylem, phloem separate, then alternate. Xylem and phloem separate, alternate, present on different radii, called radial vascular bundles. Character of roots. Then in roots, xylem differentiation is a centripetal. Centripetal means protoxylem towards outside or towards pericycle. Metaxylem towards center, or it is towards medulla. Such differentiation is called centripetal. Such condition is called exarch. So actually vascular bundles two types, vascular tissues two types, primary and secondary. Primary vascular tissues develop from procambium. Secondary vascular tissues produced from vascular cambium. Primary vascular tissues, which includes primary xylem and phloem. Rightly, primary xylem, two types, proto and metaxylem. Protoxylem, first formed xylem, it has narrow lumen. Metaxylem, later formed. If xylem differentiation is centripetal, that protoxylem outside, metaxylem inside, it is called exarch. So in roots, vascular bundles are radial and the xylem is exarch. And there is no cambium in between vascular bundles. So, it is called closed type. Then, next one is collateral vascular bundles. Collateral vascular bundles, character of stems and also leaves. Collateral vascular bundles means phloem and xylem. Phloem and xylem present on same radius. If both xylem and phloem present on same radius, called collateral vascular bundles, stems and leaves. Generally, these collateral vascular bundles, in that xylem and phloem are joined. If both are joined, called conjoint. But in dicot stem, this joining takes place through cambium. It means in between xylem and phloem, if cambium is present, it is called open type. So, only in dicot stem and gymnosperms. In dicot stem and gymnosperms, vascular bundles collateral means side by side, conjoint, both are joined, but the joining is through cambium, so it is called open type. So, collateral conjoint open present in dicot stem and gymnosperms. Then, in monocot stem and leaves, in monocot stem and also leaves, this vascular bundles collateral means xylem and phloem side by side, called collateral, both are joined, called a conjoint, but this joining is direct, means cambium is absent. So, if cambium is absent, called a closed. So, monocot stem and leaves, vascular bundles collateral, conjoint and closed. Then, in some plants, xylem present at center, xylem present at center, on both sides of xylem, there is phloem. So, xylem at center, on both sides of xylem, there is phloem bundles called bicollateral. On both sides of xylem, connected to the phloem through cambium. So, rightly it is called bicollateral and open type of vascular bundles. Bicollateral and open type of vascular bundles common in families like Solanaceae, Cucurbitaceae and even EAPCET. Example for EAPCET, Nerium. Example is Nerium. Then fourth type of vascular bundle is a concentric. Concentric vascular bundles means ring-like vascular bundles. Then These concentric vascular bundles are two types. One is amphicribal and second one amphivessel. Amphicribal is also called hadrocentric. Hadro means xylem. So, in this type, xylem is surrounded by phloem, called hadrocentric, character of pteridophytes, ferns. Amphivessel is also called leptocentric. Lepto means phloem. So, a phloem at center surrounded by xylem. It is called leptocentric. Example for leptocentric: Dracaena and Yucca. So, these are types of vascular bundles.